I think.
Yes.
If you stop,
And
then
once,
Dr. Vatsala Misra can put her slide.
can you see my screen?
No, not yet. Yes, yes. Now we can see.
Okay.
Yes, yes. Okay, okay.
So just 3 minutes more, then you can start. Good evening, Dr. Mohanty. Good evening, sir.
Sir is muted, actually.
Yes, I think so.
Sudip, you may be able to see Professor Vatsala Mishra, Professor Ashranti Kaur, both of whom you must have interacted.
Can sir please unmute?
And of course, Professor G.N. Mohanty has been a very senior and revered member of our profession, teacher, and he has steered our IAPM for a long, long time. We take his advice even now. One of our senior most member, and of course Dr. Ashim Das from PGI Chandigarh. I don't know if others have joined. I can't see anybody
We
else.
have.
Not yet.
So, Asranti, should we start?
Madam, 7, please. 3 minutes, madam.
Okay, 3 minutes. Okay, my watch. Okay, we can wait for 3 minutes.
Madam.
Okay, all the best to Sudip and
Thank you.
of course
Thank you.
Dr. Vatsala Mishra and Dr. Ashrantika.
Yes, sir. Yes, sir.
I think people are logging in. I welcome everyone for this webinar. This is third in series. Asranti?
Yes, sir. Madam, 3rd.
Third in series. We will be having many more such webinars. I welcome everyone who has logged in. I welcome Professor Mohanty sir. Then Siddharth is there. He is a past president. Then Professor Ashim Das, he is also logged in. He is also a past president. And I think that gradually all the delegates, gradually they will log in. There's थोड़ा सा problem जो login का number of delegates का है because we are having many overlapping webinars. We are having classes. The time is overlapping because there is PGI webinars are going on. Then the teaching classes of Tata are going on. Sumit and all, Dr. Bojes. And in most of them, the time is clashing. I think that is the reason here we have some number which are less who are logging in. I think as now it's already 7, so we can start with this official webinar. And I once again welcome all the members who have logged in for this third webinar in the series. And our today's speaker is Dr. Sudip Sen. Dr. Sudip Sen has done his MBBS from Calcutta He completed his MD and PhD in biochemistry from All India Institute of Medical Sciences, Delhi. He joined as faculty in biochemistry in 2012. His main research areas of interest are cancer biology and stem cell biology. He has published many papers in reputed national and international journals. He has also received many prestigious national and international awards, and mainly in the field of cancer and stem cell biology. His work has been acknowledged by various organizations like International Society of Stem Cell Research, American and European Associations for Cancer Research, ESMO Association, and UICC. He has been guiding MD and PhD and MSc students since 2013 and has received many extramural research grants from organizations like DBT, DST, ICMR, and Ministry of Ayush. So today he'll be deliberating on a very important topic, that is cell kinetics. Cell kinetics, as we all know, is very important for basic research, and mainly in the cancer field, as more and more research are now being done in the field of cancer therapy, cancer pathogenesis, cell biology is the basis of all these research. Until unless we have clear concept of these various phases of cell kinetics, then we have those concepts clear in our mind, it is very difficult to build a research work on these lines. So I think starting from UG, in UG also we teach them basics of cell kinetics to postgraduates, to research workers, or doing PhD. The cell kinetic is a very important topic which will help us in building good projects and further working and inventing newer drugs and newer therapies for different malignancies. So without taking much time, I will now hand over or ask Dr. Sudip to start with his talk. So Dr. Sudip, you can now share your screen. I'll stop and you can share your screen.
Are the slides visible? Thank you, ma'am.
Not yet. You share your screen.
Okay.
Not yet.
Sudeep, that green button down, no? Share screen.
Click on green icon, share the screen, Dr. Sudip, and
Yeah.
then share.
Huh.
Yes, yes.
Make it full screen.
Yeah.
Yes.
Thank you.
Yes.
I would like to thank Professor Misra for her kind words, and Professor Asranti Kaur and Professor Siddharth Dattagupta for giving me this opportunity to present some interesting facts about the cell cycle today. I have a confession to make at the beginning that Professor Siddharth asked me to keep it very simple and very basic, so that the concepts are made clear, as most of this listeners will be postgraduate students. So I don't want to make it very complicated or very molecular based or very complex. But as Professor Misra said, that the cell cycle has slowly become more and more important, and different aspects have been linked to the cell cycle. And in the previous webinars also, I think Professor Iyer talked about apoptosis and DNA damage and necrosis and all those things. So everything is very intricately linked to the cell cycle. Now, the cell cycle as we all know, essentially controls growth and proliferation of cells. So it regulates DNA damage, repair, response to injury. And even though it is consisting of the DNA synthesis, the two gap phases and the synthetic phase and the mitosis phase, it has got deep connections with other aspects like DNA damage, repair of damaged DNA, and many other important aspects like cancer. Now, the cell cycle, as I said, essentially consists of 4 different phases. The DNA synthesis happens in the S phase. After the S phase, there is a brief pause or a gap, which is known as the G2 phase, followed by the mitotic phase. So mitosis, we all know, is essentially where the cell divides into 2 daughter cells, and it consists of 4 different stages: prophase, metaphase, anaphase, and telophase. But what is interesting is that the DNA synthesis and the cell dividing into 2 is also interspersed with 2 gaps. So the G1 phase and the G2 phase are essentially phases in which some of the preparations are put in so that the S phase and the M phase can occur without any problems. Another very interesting thing is that when we look at the cell cycle, it appears as a very smooth and a very simple cycle in which one phase transitions into another. Now, as I just said, that G1, S, G2, and M are the 4 phases. It is also quite often these phases can be morphologically subdivided into interphase and mitotic phase. So M phase is the mitotic phase, and the interphase is all the rest of the phases, that is the G1, S, and G2. So this is known as, this whole rest of the cycle is known as the interphase and M phase. So that is also, sometimes you will see in some books it is mentioned as interphase and mitotic phase. Now, the S phase essentially is the phase where the DNA synthesis occurs. So DNA synthesis happens in different phases, and after this and before this, so before the DNA synthesis occurs, there is a G1 phase in which a lot of preparation goes on, and after the DNA synthesis, there is again some sort of recuperation from this phase as well as preparation for the mitosis phase. So these G1 and G2 phases, or even though they are preparatory phases, so-called preparatory phases, they have their own importance. Another phase which we'll be seeing is not exactly a phase in the cell cycle, but it is also called the G0 phase. So that is when cells are not actively cycling. So when the cells are within this cycle, they are said to be actively cycling or preparing and synthesizing the DNA. But when they are out of these 4 phases, they are essentially in G0 phase. They are not actively cycling, but they have the potential to again re-enter this cell cycle and start multiplying. Now, what makes this whole cycle work smoothly and without any problems is a lot of regulatory proteins. So you'll see that cyclins and CDKs are mentioned here. So from what initially cell cycle, when it came up, to now, if you look at PubMed, you'll find that there are a huge number of papers coming up every year which keep on deciphering the complex regulation of the cell cycle. Now, this cell cycle is able to function without any problems because of certain regulatory proteins. Now, these regulatory proteins are in the form of cyclins as well as cyclin dependent kinases. Now, for those of you who are a little rusty on their biochemistry, I'm a biochemist, so I would definitely like to just re-emphasize the importance of the enzymes and the proteins here, because the whole cell cycle is being regulated by these kinases. So the cyclins are essentially a family of proteins. I'll probably go to one slide where it's okay. So this is where you can see the why they are called cyclins. So the name itself is given because these proteins appear at different points in the cell cycle. Their cyclical dynamicity is the reason why they are called cyclins. And you will see that here, obviously, there are just a few cyclins given. But if you look into different papers that have come up, many more cyclins have come up. And within a group also, there are subgroups. So I don't want to make matters unnecessarily complicated, but we'll keep it very simple. So these cyclins are some proteins which appear at specific times in the cell cycle. Now, these cyclins, they activate cyclin dependent kinases. So if you remember, the kinases are enzymes which phosphorylate, and the opposite of kinases are phosphatases. So they dephosphorylate. Now, again, going back to basic biochemistry, enzymes can be activated and deactivated. Proteins can be activated and deactivated through different mechanisms. Phosphorylation and dephosphorylation are some very important and very basic mechanisms by which different proteins are activated as well as deactivated. So cyclins are essentially proteins which activate the cyclin dependent kinases, and these cyclin dependent kinases, by virtue of their kinase activity, they phosphorylate different proteins. Now, it is commonly a mistake that people believe that phosphorylation will always activate a protein. It might not be so. So sometimes you will find that phosphorylation can even deactivate a protein. So something which is normally active can be deactivated by phosphorylation. So it is not always that phosphorylation will activate a protein. So these kinases in different phases are activated by these cyclin proteins. Now, another very interesting thing is because of their cyclical nature. So you see that these cyclins appear at specific times. So this is the mechanism by which these cyclins make sure that a particular set of cyclin dependent kinases are activated. So you can see that some cyclins activate some kinases, and so it is not always that one cyclin dependent kinase will only interact with one cyclin. It might be multiple interactions also, but essentially the regulation is taken care of by the cyclical or the dynamicity of the cyclins or appearance of the cyclins. So these cyclins, the appearance of these cyclins makes sure that only specific cyclin dependent kinases are activated during specific phases. So you will have cyclin E active during the S phase, cyclin A coming up. So when one cyclin, the level of one cyclin falls, the other cyclin is rising. So one activity is giving way to another activity. Now, how is this regulation made possible? So again, even though this is a pathology and microbiology webinar, I being a biochemist, I have the this thing of pulling back everything and making everybody believe that everything is based on biochemistry. So please forgive me for that. But essentially, the regulatory aspects are actually maintained by biochemical reactions, which are very simple, but which have a very important role to play in maintaining or the activity of important proteins like the cyclins. So for example, so let me give you an example of what I was saying. So cyclin D. So cyclin D, let me go back to, okay, here. So cyclin D actually, see if you can see it, it is interacting with cyclin dependent kinase 4 and CDK6. So these form a complex. So you can see from this that this cyclin goes and attaches to the cyclin dependent kinases. Now, essentially, these cyclins have a very interesting role to play, that they, by binding to the cyclin dependent kinases, they activate these cyclin dependent kinases. So the cyclin dependent kinases by themselves are not active. So there comes one layer of regulation, that one, as the cyclins go and bind to these cyclin dependent kinases, they activate these CDKs. Now, another very interesting thing is that these CDKs lack nuclear localization signals, which are again provided by the cyclins. So the cyclins bind to the cyclin dependent kinases, activate them, and transfer or translocate them to the nucleus so that they become active. And as I said before, some cyclin dependent kinases bind to more than one cyclin. So it is not that it is always a 1:1 corresponding ratio. And the cyclin dependent kinases, as I re-emphasize again, they can be activated only at specific times during the cell cycle because of the cyclical activity of the cyclins. Now, cyclins have another very interesting phenomenon, is that they undergo hello.
Can you mute Dr. Kar?
Okay, I'm sorry. So cyclins, we were talking about how cyclins, the cyclical activity of these cyclins arises. Now, these cyclins undergo regulated degradation. So these levels rise and fall by virtue of certain sequences in their protein structure, and they have a sequence called PEST sequences. So the P stands for proline, E for glutamate, S for serine, and T for threonine. So it's basically a PEST sequence which targets them for degradation. So these sequences are activated and they are regulated so that the cyclical activity of these cyclins is maintained. Now, coming back to our CDK4. Now, as I said, so I'll give you an example of how the activity is regulated. So I told you that cyclin D activates CDK4 and CDK6. Now, after they form a complex and become activated, this whole unit, cyclin D plus CDK4 and cyclin D plus CDK6, it starts to function as an active kinase. So it's an active enzyme. Now this whole unit is an active enzyme. Now it looks for a substrate. Now what is the substrate for this? CDK4 and 6. It has, among other substrates, a protein which you might have heard of is called the retinoblastoma protein. And what do we know about the RB protein? If you remember, the RB protein or the retinoblastoma protein is a very important tumor suppressor uh, gene or a protein. So along with that, the retinoblastoma protein has also a very important role to play in the cell cycle. So I'll just come to how retinoblastoma or RB protein is regulating the cell cycle. So as I said, this active kinase enzyme or this kinase unit acts on retinoblastoma protein. So I have a picture here. Okay, so this is what I was talking about. So this whole unit, the cyclin dependent kinase 4, and with the cyclin D and cyclin dependent 6 with cyclin D, this whole, there are actually two units here. The picture is showing it as one. This acts on the RB protein and phosphorylates it. Now, as soon as the retinoblastoma protein is phosphorylated, it dissociates from a transcription factor called E2F. So it's previously there as a unit, and as soon as the phosphorylation happens by virtue of this kinase, RB gets phosphorylated and E2F gets released, and the inhibitory role of retinoblastoma is lost. Now, this transcription, this activates transcription and ensures that cell cycle progression takes place. So this is how beautifully the regulation happens. So actually it looks very complicated, but it's a very smooth and a very beautiful regulation which happens. The cyclin increases. As the cyclin increases, it gets binds to the cyclin dependent kinase, activates it, moves it to the nucleus, and these become as active kinases. They bind to their substrate, phosphorylate it, and as soon as the phosphorylation happens, these units dissociate from each other, and the phosphorylation of retinoblastoma results in release of E2F from the transcription repression which RB was doing. So this transcriptional repression is released by this phosphorylation, and E2F, the transcription factor, is allowed to progress, and the cell cycle progression takes place. So this is how very intricately this regulation happens. Now again, going back to, so I will just like to emphasize on 2 or 3 cyclins and how they act very briefly. So the next is cyclin E. Now cyclin E is again, so if you notice that cyclin D was happening in G1, and when this activation happens and when this progression, so the repression of transcription which was happening due to retinoblastoma protein, as it is removed by the phosphorylation, this moves from G1 into S. Now there is, you will notice there are certain checkpoints which make sure that this is ready to go into the next phase. So as this transcriptional repression is lifted, it passes through the G1 phase checkpoint and enters the S phase. Now in early S phase, cyclin E and CDK2, they form a complex in late G1 or in early S, but cyclin E is rapidly degraded. So let us see how fast cyclin E is. So you can see that cyclin E starts appearing in late G1 and is rapidly degraded. And as soon as cyclin E is degraded, another cyclin called cyclin A appears. So this is how intricately or how beautifully the coordination between cyclin E and cyclin A happens. And you will see that this unit gives way to this unit. Now cyclin E is degraded and CDK2 then forms a complex with cyclin A. and this is needed for the initiation of DNA synthesis in S phase. So this is how the S phase checkpoint is overcome, and then it goes into the G2 phase. Here again, cyclin B and CDK1, after cyclin A is done away with, after it is ubiquitinated and degraded, cyclin B comes in, and cyclin B combines with CDK1 to form a complex and ensures that the G2M transition takes place. So I'm not going into the very detailed intricacies of the mechanisms, but just to emphasize that this is how the cyclins, by virtue of their cyclical appearance, activate the cyclin dependent kinases, and these kinases, by phosphorylating certain proteins, activate them or deactivate them, by virtue of which the cell cycle progresses. So this is very simple but a very interesting story. Now coming to the checkpoints. Now the checkpoints, as you know, the name suggests that these checkpoints are very important, not only to make sure that everything is working properly, it also weeds out certain damaged cells which do not clear the criteria for moving into the next phase. So for example, G1 checkpoint looks at cell size, the nutrients, growth factors, DNA damage, and if the stringent criteria are not met, then it will not allow the cell to go into DNA synthesis. It may just go out into G0 phase, or it might go into apoptosis through different mechanisms. So these are stringent checkpoints. One is, so there are 3 or 4 checkpoints. One is in G1, so that I told you looks for certain criteria. Another checkpoint looks in G2, looks at cell size and DNA replication and checks and keeps those cells which are not meeting the stringent criteria from entering into mitosis. So these are again protective mechanisms by which you make sure that something which is incomplete or not ready to proceed into the next phase does not enter the critical phase of mitosis. Because you know, DNA synthesis, mitosis, these are very critical phases, and if mutations occur, then obviously cancers can happen. And in the M phase, there is a spindle assembly checkpoint which looks at chromosomal attachment to spindles. And as I said, the different phases, prophase, metaphase, anaphase, and telophase, how the spindles separate and how the, so those mechanisms, there are again checkpoints or stringent mechanisms by which the defective cells are not allowed to proceed further. So these are very interesting mechanisms. And we will find that in certain diseases, in cancers, these checkpoints are somehow overcome so that defective cells are allowed to move through. So we'll see how that happens. Okay. If you have any questions, please feel free to interrupt me and ask. So we talked about the different phases. We talked about the different checkpoints. This is how the different cyclins and the different proteins, the regulatory proteins, appear and disappear. So you can see the S phase cyclin, the G2M phase cyclin. So these are different cyclins. We talked about cyclin A, E, D. So these are again different cyclins which appear, bind to the CDK, the cyclin dependent kinases, activate them. And so this is the cyclical story as the cell cycle progresses. Okay. And if you remember your basic biochemistry again, you'll remember that each different enzyme has specific substrates. So every cyclin dependent kinase, the active cyclin dependent kinase, which is active enzyme which is formed, will have different substrates which it will phosphorylate. Okay, now this is what I was talking about, the cell cycle checkpoints. So these checkpoints if entrance into M is blocked, and if DNA replication is not complete. So these are again checkpoints, as the name suggests, that if they are not met, if the cells are not meeting the stringent criteria, then these checkpoints will be blocked and the cells will not be allowed to enter into the next phase. Now, what is interested is that these checkpoints are disrupted in some way or the other in cancers, and we will see exactly how that happens. Now, these are a few interesting cartoons which I found, and which will again emphasize that what we were just talking about. So just recapping what we talked about. So here there is no cyclin, there is the cyclin dependent kinase. By itself it is inactive. It cannot target or phosphorylate the different targets because it is inactive. So because there is no cyclin. Now, as soon as the specific cyclin comes and binds to the cyclin dependent kinase, the kinase phosphorylates the targets, and because it's a G1S, so the S phase begin and its targets get phosphorylated after the whole enzyme is activated. So this is how the regulation takes place. So it's not that anything can start working on its own. There is a very intricate and complex mechanism which makes sure that nothing just starts working on its own. So only after the cyclin appears, it binds to the cyclin dependent kinase, does this CDK become active and the phosphorylation occur so that DNA replication enzymes are activated. So this is how the S phase activation. Now this is again the mitotic cyclin. So which was the mitotic cyclin? Can we just go back and see? In M phase, the cyclin B. Okay. So here the M cyclin or the cyclin B goes and activates this cyclin dependent kinase, and as soon as it is activated, this whole cyclin dependent kinase starts phosphorylating its different targets, and M phase begins, and spindle formation, chromosomal condensation, nuclear membrane breaking down can happen because the M phase targets are phosphorylated. So all the mechanisms are essentially controlled by these cyclins and cyclin dependent kinases. The intricate mechanisms happening in mitosis or happening in during DNA synthesis are controlled by these cyclins and cyclin dependent kinases. Now, another very interesting thing, all of you must have heard about P53. So P53 is known to be a very important tumor suppressor gene. It is all sometimes called as the guardian of the genome. So, and it screens for DNA damage. And I'm sure Dr. Iyer must have talked about its role in apoptosis and how it detects DNA damage. So we'll be just shedding a little light on P53. Now, what P53 does is that after it is activated, it is, it's not a very stable protein. It's quite an unstable protein, but it, as soon as it detects DNA damage, it somehow gets more stable. The stability of the P53 increases, and because of the DNA damage, and the P53 thereby activates a certain molecule called P21. Now this is a very important molecule because P21 is a potent cyclin dependent kinase inhibitor. Now this, being activated by P53, goes and binds to this active enzyme unit, which was activated by the cyclin and the CDK kinase binding together, and inactivates it. So it leads to the G1 arrest, which is associated with P53. So this is another very interesting mechanism which happens in P53 or when it works or when it doesn't work. So when there is DNA damage, P53 is activated, it binds to the DNA and it halts the G1, the cell cycle, and DNA repair is initiated. And if DNA repair is not possible, then apoptosis is triggered. And so that cells don't pass the damaged DNA. Okay. Now, non-functional P53, if P53 is mutated or if there is any problem with the P53, then P53 cannot initiate these activities, and because of that, the cyclin dependent kinases and the cyclin remain active, and the cell keeps going into cell cycle again and again, and ultimately the mutations are passed on. The checkpoint is DNA repair does not happen, apoptosis does not happen, and the cell cycle continues to progress, and the damaged DNA continues to mutate and cancer sets in. So this is a very simple cartoon which explains how P53 acts and how the P53 by virtue of P21 inhibits the cell cycle progression and causes a G1 arrest. Now here again, you will see the different proteins P21, P57. There are different proteins, not only P53, but different proteins which are in turn regulating the cyclin dependent kinases, the cyclin dependent kinases, which we talked about. So this is not just to complicate the mechanism, but these are nature's way of devising backup systems and complex interrelated backup mechanisms so that nothing is allowed to function without something controlling it from behind. So these cyclins are inactivated by certain enzymes. These are again linked to certain other proteins. So it's a very complex mechanism which is controlling the cell cycle so that it suddenly doesn't start working on its own and develops a mind of its own and keeps on proliferating and the cells go into cancer. So this is what we were talking about, the E2F and the retinoblastoma protein. And so this is what we had talked about, the S phase entry and E2F dissociating from the inhibitory effect of retinoblastoma, allowing the S phase to function. So similarly, you will find that P53, I told you, has the ability to go into apoptosis or go into cell cycle arrest. And there are CDK inhibitors which are happening not only for one specific CDKs, but different inhibitors, and MDM2. So you know that P53 activity is again regulated by MDM2. So MDM2 is again regulated by different mechanisms. So it's a very complex interrelated crosstalk which is happening, and this different layers of regulation makes sure that nothing is allowed to run away and start developing a mind of its own and become a loose cannon. So everything is very intricately regulated through some mechanism or the other. So here you can see the mitogenic signaling, which we are familiar with, the RAS, RAF, MEK, ERK. You are familiar with these mitogenic signaling pathways. These are again in turn controlling the cyclin G1. And so we didn't talk about cyclin G too much in detail, but this again acts to control MDM2, which is in turn controlling P53. Yes, Prashant. Prashant has a question. Can you unmute yourself and ask the question? Or is it on the question? No open questions.
I think you can continue, Dr. Sudip.
You
Okay.
can write in the
Finish
chat
the,
box.
in the end, we
At
take the question
we
answers.
can take
Yeah.
the questions.
End.
Okay, so we have covered these parts, and let us move a little ahead. And so we talked about how cell cycle regulators, so we talked about the checkpoints and how these proteins or these molecules are preventing cancer from happening, and they are normally functioning. So p53 promotes G1 arrest. We talked about how p53 causes G1 arrest after DNA damage and does not allow the cell to go ahead. And we saw that if it is mutated, then this arrest does not take place. And p53 mutations, I'm sure all of you know that very many tumors are associated with p53 mutations. That is why it's called the guardian of the genome. And if p53 is mutated or is defective, then it can very often lead to cancer. And we also talked about how retinoblastoma is a restriction point regulator. It has an inhibitory effect, and that inhibitory effect is obviously, we were talking about how that inhibitory effect is removed and the cell cycle is allowed to progress. But that inhibitory effect, if it is totally lost, then again it can lead to cancer. So that is why retinoblastoma is, even though it's an inhibitory protein or an inhibitory gene, but its deletion can allow cancer to take over. So here again, MDM2 is intricately linked to p53, and it overrides the p53 transcriptional activity. And in case there is a loss or a gene amplification or enhanced mRNA translation, then it leads to oncogenesis. So similarly, again, p16 is another protein which inactivates G1 cyclin CDK complexes, and this inhibitory effect, if lost, can again lead to cancer. So we looked at cyclin D1 also, and cyclin D1 acting with CDK4 and CDK6. And here again, if it is overworked gene amplification, there have been certain cases where gene amplification have led to cancer or oncogenic transformation. Again, these are again cyclin dependent CDC cyclins, which are phosphorylated and which activate the cyclin dependent kinases. So these are also associated, if overexpressed, they are associated with cancer. So these are just a few examples of commonly associated proteins which can lead to cancer. Now, going ahead. Now, this is a very interesting and a very important thing which I think all of the people who are attending today's webinar should definitely know, is that how do we evaluate the cell cycle? So I think most of you are pathologists, so I probably will not need to talk about immunohistochemistry, and a biochemist explaining immunohistochemistry to a pathologist is, I think, an absolute no-no. So you are all familiar with, and I think you know immunohistochemistry much better or much more intricately than me. So just to focus on these markers. So these are the cell cycle markers which are commonly looked at. So Ki67 and PCNA are, I'm sure you are aware of these two proliferative markers, the cyclins we talked about. So these markers will tell you about how and is there any overexpression or underexpression. So the proliferative index being given by Ki67 and PCNA. Cyclins, obviously not a specific cyclin, but different cyclins will give you an idea of exactly which part is normal or abnormal and things like that. So here I would just like to bring your attention to two things which are probably you are not very familiar with. One is the BRDU or bromodeoxyuridine assay, and nowadays thymidine is not that much followed because it's a radioactive technique. So BRDU is, you will often find in a lot of papers, BRDU assay, bromodeoxyuridine assay, and assessment of ploidy. So this is flow cytometry and this is immunohistochemistry or flow cytometry, but I'll just briefly tell you the principle of these. So in flow cytometry, I think I have another 5 minutes or 10 minutes.
Yes.
Okay, thank you, ma'am.
Yes, yes, you can go on. You have, yeah.
Yeah. So DNA ploidy is essentially looking at the cell cycle, and there are fluorescent markers. Propidium iodide is used that binds to the DNA, and in a flow cytometer, laser lights illuminate this propidium iodide, and it can be detected that which phase of the cell cycle the cells are in. So this is how a normal cell cycle, G0, G1, S phase, G2, M phase. So these are the number of cells which are in this phase, and this number of cells. So obviously some cells will be in S phase, some cells will be in G0, G1 phase, some cells will be in G2M. So you will find this is usually a normal looking peak. Now, if there are certain arrests, if there is an arrest here, slowly the cells will decrease, this cell will, so the by knowing the normal, you can evaluate when there is a arrest or if there is something wrong. If you can actually look at apoptosis, you can look at G2M arrest, you can look at S phase problems. So you will find numerous images in different papers which look at which analyze derangements in the cell cycle by using flow cytometry and propidium iodide. So this is how it looks, and softwares are available which can tell you exactly how much percentage of cells are in each phase of the cell cycle, and if there are any derangements you can find out. Similarly, if you are giving some drugs to some cancer cells and if you want them to arrest in a particular phase, you can also assess that. So this is a very interesting technique, flow cytometry. Now, BRDU incorporation assay is also something which is based on the fact that bromodeoxyuridine is an analog of the DNA precursor thymidine. And because of that, when in culture media, if you incorporate BRDU, then it goes in the replicating DNA and can be detected by using anti-BRDU antibodies. So in proliferating cells, DNA is replicated, and when BRDU is added, it is incorporated, and as because it resembles thymidine, it is incorporated into the DNA, and the amount incorporated can be assessed by anti-BRDU antibodies which have a fluorescent tag. So again, like flow cytometry, you can look at different phases of the cell cycle and assess if there is any arrest or if there is any decrease or increase in any particular phase compared to the normal. So these are the BRDU and the ploidy. Thymidine, as I said, again, very similar, but they use autoradiography, but nowadays not much preferred because it's a radioactive technique. And Ki67 and PCNA I talked about. So these are essentially looked at by flow cytometry or immunohistochemistry. So immunophenotyping is done in flow cytometry, and immunohistochemistry, primary antibodies and secondary antibodies tell you the level of expression of these proliferation markers, which can tell you. Now, cyclins we were discussing at length. So if any particular cyclin is increased or decreased, then it can give you a very good idea about whether it can be whether it has been deranged and things like that. So these are again markers and techniques by which you can evaluate the cell cycle, and if you are giving any drugs, then you can also evaluate the effect of those drugs. So this brings me to the probably the last part of today's talk, and this is a comparison. So you can see the upper portion are the drugs which most of you will be familiar with. So they are the chemotherapy which we have most commonly used. So cisplatin, carboplatin, temozolomide, ifosfamide, busulfan, topotecan, irinotecan. These are commonly used anti-cancer drugs which are commonly used in the clinics, and paclitaxel, vincristine, vinblastine. So these are again, I'm not going into the mechanisms because we are very familiar with how these drugs are acting. But what is interesting is that now this chart from American Association for Cancer Research also includes the newly developing drugs, which we talked about a few minutes back, cyclin dependent kinases being acted upon. So see, we talked about CDK4 and 6. And so there are drugs which are acting on these cyclin dependent kinases, and whether it is specific cyclin dependent kinases or pan-cyclin dependent kinases which are affected by these drugs, and these ensure that the malignant cells or the proliferating cells are blocked so that the cells don't proliferate in an unregulated manner. Okay, so flavopiridol, it's again I think a semi-synthetic flavonoid derived from a plant, and so these are all experimental drugs which are looking at ataxia telangiectasia mutant. So this is another kinase which is checkpoint kinases. So these are very important regulatory proteins which are being targeted by these drugs. And again, these are cyclin dependent kinases. So if you look at some papers, I didn't talk about V kinase because I didn't want to complicate things too much. But again, these are kinases which in turn regulate the different cyclins or the cyclin dependent kinases. So these are again also possible targets which allow the cells to be prevented from moving forward. Now, these are again serine threonine kinases, aurora kinase and polo-like kinase. These are involved in mitosis, and these are serine threonine kinases. And also newer drugs are being developed to target these serine threonine kinases so that they do not allow the cells to proliferate. So these cells, by virtue, these drugs, by virtue of their specific activity, have much less probability of side effects. But obviously, they are still in the experimental stages. And I think when these drugs come into the clinics, then we'll exactly know that do these cells also have any deleterious effect on the normal cycling cells. So that is something which we all hope that these will only be specific for the cancer cells, but till you actually give them to patients, you will not know what is their role. So probably decreasing the activity to a certain extent, to a baseline extent, is possibly my understanding how this will prevent. So this is again a cartoon which shows you how different mechanisms are preventing the cyclin dependent kinases from functioning normally. So whether it is imidazopyridines or flavopiridol, which we talked about. So these are how the active kinases are prevented from functioning normally. So I think I can wind up my talk. I think my purpose was not to impress upon you or to overwhelm you with data or with newer molecules and complex mechanisms, but just to simply tell you how the cell cycle happens, how it is regulated, and what are the mechanisms or what are the techniques by which you can evaluate cell cycle disruptions, and the newer drugs which are coming up, which exploit cell cycle mechanisms and are less toxic than the currently available chemotherapeutics. So I think I'll finish here, and any questions which are there, I would be happy to take questions. Thank you.
I think one question was from this Prashant you were telling, no? He had asked one question. What was that question, Prashant?
I think he had raised his hand either by mistake because he didn't ask there.
Yes.
And thank you, Dr. Sudip. Any of the delegates, if you have any question, I think we cannot see. There is no question in question answer also.
So I was either very bad or
No.
very good.
Very good.
No.
It was very good. And I think it is really very important. I feel it's very nice.
Yes.
Because basics of, as I said, the basics of cell kinetics are really very important. If you are working as an oncologist, if you are working as a pathologist, if you are working as an oncosurgeon or co-physician, because when you are using these drugs, at that time you have to know how they are acting. Yes. And where they are acting and what is their targets. And until you understand the basics of cell kinetics, you won't be able to understand their effects. So this topic was very good, and he explained it very well in a, can say, simplest form, that how these changes are taking. Now PGs, they keep on mugging up. इसमें CDK4 express होता है, इसमें CDK6 होता है, इसमें CDK2 होता है. They keep on mugging up those, and that's why they cannot remember, cannot understand. If you understand the basics of this cycle, as has been very well explained by Dr. Sudip, I think then you will not need to mug it up, because you will understand where it is acting and where, how it effects can be seen, or where you can see their effects in different tumor cells, and similarly for diagnosis or similarly for treatment. So I'll say that this was an excellent lecture, very well explained in simplest form the cell cycle, cell kinetics.
Thank you.
I
Thank you.
myself was very afraid of cell kinetics from when you start teaching.
Yes.
Yes, madam. Yes, madam.
Yeah.
When we have to upgrade ourselves with all these new markers which are coming day by day.
Yes.
So if you understand the cell cycle clearly, so I once again thank Dr. Sudip. Is there any other comment, Dr. Siddharth Dattagupta?
No, no, I think he has spoken very well and in a simplified manner
Yes.
explained to us what the
Yes.
cell cycle is. Most of the time we are in the dark actually. So
Yes.
we just stay in and see. But
Yes.
after this, we'll be able to understand why we are doing things.
Yes. Dr. Ashim Das, any comment? Your comments, please. Dr. Ashim, can you hear me?
I think he is not able to hear, but
Yes.
I think this is what was actually necessary.
Yes, I also agree
Simplified
with
and
sir
very
and
nicely
madam.
done.
Yes, sir.
Yes. Yes.
Very good, Sudip.
Very simple. He started from very simple and went to two difficult areas. And the best part was the difficult techniques and markers he put in a table to know the cell cycle derangements. That is very essential for all of us. That was for the pathologist point very good. So I also thank Dr. Sudip.
Thank you.
There is
Yes,
one.
there is a question from Dr. Kamalini Mahapatra. Is there any lab in India to study the cell kinetics? Dr. Sudip.
Ma'am, a lot of people are working on cell kinetics and looking at different aspects. So all these markers are being studied in many different labs. So whoever is actually working on cell kinetics and cell cycle related markers will definitely be doing some of these assays, and these are not very difficult. I am sure as pathologists you are perhaps the best people to tell us about immunohistochemistry, but BRDU assays and flow cytometry nowadays in most advanced or in
No, flow cytometry and immunohistochemistry, we are doing it regularly. But BRDU assay, where it is being done?
Ma'am, BRDU assays, these are again kits which are available, and
Okay.
BRDU is you just have to buy a BRDU and BRDU antibody, anti-BRDU antibodies, and this can be done by flow cytometry also. So
Oh,
you
flow
just need a flow cytometer and a kit which can tell you about. And the best thing is that it's non-radioactive, so it's very easy to do.
So I think, Ashanti, there are no more questions.
Yes, madam.
In fact, earlier, madam, we used to do the thymidine, tritiated thymidine assay, 3H thymidine, but
Yes.
that was radioactive and that had to be developed and so on and so forth.
Yes,
Now
that
I
was.
think BRDU has become easy.
Yeah.
Hello.
We are doing.
ମୁଁ କେମିତି କହିପାରିବି?
ସେ କୁହନ୍ତୁ ଆପଣ କୁହନ୍ତୁ ସାର୍ ଏବେ କୁହନ୍ତୁ
ହଁ କେମିତି କହିବି?
ସେଇଠି କୁହନ୍ତୁ ସାର୍ କୁହନ୍ତୁ ସାର୍ କୁହନ୍ତୁ ସେ ସ୍କ୍ରିନରେ କୁହନ୍ତୁ
just I want when I entered in the MBBS in the first year 1961. My teacher in biochemistry came from All India Institute. He was with MSc biochemistry, and he always start telling the life is nothing but series of biochemical reactions. So at that time, biochemistry is nothing, and we are taught with organic chemistry. And that in 1961, my teacher from All India Institute, and today Dr. Sen from All India Institute. So within the 60 years time, I find there is a great change and advances. Somehow he made it so simple so that I could able to understand it broadly what I come
Yes, sir.
across, and that's a very good. I must congratulate him making such a difficult for situation as A to Z of cell kinetics. I was very much astonished how it will be complicated. Thank you, Dr. Sen, for your
Thank you, sir.
nice.
Sir, it's a honor coming from these words coming from you, sir.
Yeah.
Yeah. So thank you, Dr. Sudip.
So
And
thank
about
you.
the next webinar, I'll discuss with Vatsala ma'am and come up, because that also is again on general pathology like hemodynamics. So should we have that, or should we shift to some this like slide seminar, which will be more interesting?
I think
So,
in between, Saranti, we can shift to some slide seminar.
yes, madam, because like continuous they will not be interested, the students.
Yes. So
Okay.
we had a basic, it was let them assimilate all this and understand, and then we will talk to slide seminar, and then again
Yes,
move to some basic topic.
madam. So I'll notify that in by mail or WhatsApp.
Okay, okay, Asanti.
Yeah. Asim Das sir has